Okay, I'd like to welcome everybody to the July 22nd meeting of the IMAG MSM Working Group on Multiscale Modeling and Viral Pandemics. This is our middle of the summer session. Appreciate you being here. As I need to remind you, this meeting is live streamed and recorded, and those recordings are available to the public. As always, you have access to me and Reinhard if you have any suggestions or comments, and to Jim Sluka, especially on the web side of things, and to Bruce Shapiro, anything for communications that you want to have taken care of. We're always available to help. We have our usual communication channels. Please help us make them work. We have lots of great YouTube videos, and we have a LinkedIn page, and of course there's the IMAG wiki, which is a little harder to use in terms of editing. There's a Twitter feed and a Slack channel. Please do help us let people know about these meetings and take advantage of the video archive that we have. Are there any short announcements that people would like to make this week? John, did you have anything you wanted to say this week?
Not particularly. We've gotten some input, and I'm trying to put together a package now to send up to NIH to see what we can get incorporated into a meeting sometime late fall. I would still invite any of you that have a topic that you would like to have discussed, or that particularly anything you would like to take on the effort to put together a session on a topic. Probably at this point, best to just send me an email direct, because we're real close to having to put in something and see what happens.
Is that document still live from the Google Doc from last week or a couple weeks ago? Is that the reference point?
I know it's still there.
Okay. Is that kind of the state of the state, or is there something else?
So I was hoping that in tomorrow's steering group meeting, John could give us an update on where we are.
I just wanted, he had said he wanted to announce that request input, so I wanted to let him have his.
Yep, and that tomorrow's steering group committee is kind of be the final decision point here, to probably go ahead and just submit what we have and see if we can sell some of it.
Okay, so upcoming meetings, we need your help. We have the 29th is nicely filled. Gary is going to speak, and Keisuke Ejima is going to speak, which is great. We do not have speakers lined up for August 5th. We have one speaker for August 12th. We would beg everyone to suggest at least one name of somebody who would like to hear. That could be an experimentalist. It could be somebody developing experimental technology. It could be a clinician, somebody with data, or a modeler. Feel free to suggest yourself as a speaker. Feel free to suggest your graduate students or postdocs if they need a moment in the sun. Please, if everyone sends us one name, we will fill our list. Try to be creative in your thinking. People who are doing things analogous to what we do in related fields, medical diagnostics, drug development, any of these things would be appropriate. Please send those emails to me, Reinhard, and Bruce. Bruce is extremely efficient at getting people on the schedule. So please do help us keep our schedule full. We have quite a few speakers lined up for the fall, but for August, we are short. So we would very much appreciate that. If people are in working groups and would like to use a whole session to discuss working group issues, that's fine too. If you're a working group leader and you would like to report on your subgroup's progress, that's also fine. So please let us know how we can use these Thursday slots to be most useful to everyone. Reinhard, did you have anything you want to add on that?
No, thank you.
Okay. Okay, so for the meeting, please hold your questions till the talks are over because they're short. Please do mute when you're not speaking. And occasionally people will accidentally annotate the speaker's screen. So please don't do that. We leave the annotation turned on because it's convenient for people to use it, but that means you have to be a little careful about that. If we need them, we will have breakout rooms after the meeting for additional discussions. And I will give people a 5-minute warning if it looks like they're going to run over. As you know, I do not generally give an elaborate introduction. I think we're all familiar with the recent review article that Angela did. And so without wasting any more of her time, I'd like to turn it over to her.
Thank you all. Thank you for the invitation. I have a little bit of a, just shout out to some of the stuff in that review and summary, and then a little bit of new stuff at the end. So I'm going to get my share screen going here, make sure everybody can see it. Good. So this group, this slide is probably a little bit of beyond review, right? So respiratory infections, lung insults are, among the top causes of death worldwide. A lot of us use mathematical modeling to attack these problems. When my graduate student was, doing her lit review on these, we found that it might be very useful to have a review that kind of brought these together and like systematically went through this and tried to kind of look at what were some of the similar findings from these techniques. So, sorry. That is what gave rise to this paper, which has some of the most elaborate tables I've ever been involved with the creation of. So my student also made this figure and drew everything on it. I have to give a major shout out to Sharon Minucci, who now is at Applied Biomass. She recently graduated. But you'll see the section marks here, just kind of giving you an idea of what is going on. We're giving a general overview and then kind of grouping everything into infection, focusing on influenza and pneumonia. And then also we had a section that's focusing more on active versus latent infections, so TB and anthrax, and then non-infections, so like some of your asthma type things or inhaled particles. That had a lot less papers, and that's actually kind of near and dear to my heart of what we've been doing lately. So also within there, we tried to pick out models that to use as examples to illustrate some of the approaches that were going on, but summarized a lot of the different approaches. So you'll see that there's agent-based modeling and other PDEs, ODEs. And as I said, this gave rise to, this is just, I think, a third of one of 5 tables. But what we did was pick out the key variables, key parameters for each of these papers, and then also what type they are. So you'll see it's ODE and then more hybrid models and whatnot. And we started with a some examples. This one is an influenza model. It was able to capture dynamics from 4 different strains from Manchanda et al. But in general here, you'll see I try to group some of the results from all of the influenza models or all of the ones in this section, which were, there was also separate tables for pneumonia and whatnot. And in general, there was an emphasis on adaptive immunity, which was not surprising, but that was also being found mathematically important in these models too, the parameters associated with this. And almost everybody was, or I shouldn't say most, many people were saying that co-infections were what they wanted to look at next with their models, a lot of future directions in co-infections and whatnot. I actually heard Amber Smith give a talk recently on co-infections. I think she talked right before I gave a similar talk. So Also in the latent versus active, we focused on Dinesh Krishner's labs, ABM, Graham Sim. One of the iterations, this one I think was maybe the second iteration. And again, this gave like the more of a multi-scale, multi-compartmental approach, which again, this group is probably very familiar with those approaches. And a lot of the groups use those methods, and they, as far as the immune system side of things, as far as the host response, a lot of it was the phagocytosis by macrophages, which given the nature of these latent diseases was not particularly surprising. And then, but sensitivity analysis in many of these models was showing a huge dependence on, or I shouldn't say huge, a significant dependence on bacterial replication and decay rates. So this is how, you know, I'm just kind of giving you, I hope it's a good resource for people to use, and it's just a nice place to be able to click on a bunch of links to other papers that fall into these different groups. The last group here is related to non-infectious injuries. A lot of these have a very similar regard, and this example here is from Brown et al., and it was looking at inhaled particles and how they gave rise to inflammation. And they do claim that, you know, TNF alpha and TGF beta 1 are more like lump parameters for similar type cytokines, and there was no actual data and stuff related to this, but a lot more of the repair process that goes in when that damage. A lot of the inflammation triggered is the same because it's damage to the same lung, and there's no infection, so there's not much in the way of, you know, maybe something specific to influenza or whatnot. What I do is related to this because of the lung damage side of things. So I've looked at modeling ventilator-induced lung injury for many years now, and I always, almost every paper I write, every grant, everything, I have to explain why I look at this without the cause of getting on the ventilator. And a lot of that, right, because what we're focused on right now is the damage induced by the ventilator, which is going to be different depending on why you got on there, but also we wanted a better understanding of the underlying damage caused by forcing that air into your alveolar space and triggering those epithelial cells to send out signals. So I work with a biomedical engineer who does stretch experiments and does animal experiments where the only thing that is causing their lungs damage is the ventilator. So we look at ventilator-induced injury, and of course, in the long run, to make this more translational, it's going to have to include while you're on the ventilator. But what we're seeing in mice and what we see in humans, and which is seen during COVID, is the fact that this is age dependent. So that's why our little ventilator guy there has some gray hair. Children tend to do pretty well on ventilation, but the older you are, you know, there's increased mortality. So what we then are doing is focusing on using a multi-compartmental model. Our collaborator was looking at macrophage phenotype, whether it was an M1, M2, and how that more, and we were thinking about more how like maybe the baseline relationship between these different cell types gives rise to a different response. Like if you're older and your baseline is different than younger, right? That's a state that you would have before you went on the ventilator. I'm not thinking of differences that may be caused by the underlying reason that you're going onto the ventilator here. So to do that, we used sampling. We did reduce it down significantly by requiring that we start from a steady state, because we did want to look at what was being induced by turning on the ventilation. We did not want to see dynamics that were mainly because we had started away from the fixed point and we were just moving towards it. So starting from a steady state, numerically determined steady state. We don't have any, nice formulas for how to get steady state in these cases. We then simulated ventilation. Many of the simulations looked like, and it's only 2 hours here because we were dealing with mice. Long term, we want to tie this into our data from both age groups, but for right now, we were just simulating the whole population with ventilation, regardless of whether the initial conditions were in line with an old mouse or a young mouse, but we were still doing 2 hours. But there are cases where you end up post ventilation in a different outcome, right? And so when a long-term question that my student addressed in a different project was, when can you predict correctly that that person is going to do worse and needs intervention? And so that was, very simulation based and led to, as you can imagine, many unanswered questions in more questions, I shouldn't say, led to more questions as we were trying to answer that. For one particular example, we started to think about many different ways you could come after that problem. But in this, we just first determined what was in steady state, starting from a couple of different initial condition scenarios, ran it out, and we did some classification if after ventilation you had a severely damaged lung, and then also after a recovery period. So that's that 2 hours versus 200. What you're seeing in the parentheses is those that change state. So they might have started healthy. They might have had an initial condition that is associated with a steady state of moderate damage or severe damage. And then once the ventilator happened, right, the severely damaged didn't suddenly get better with ventilation, which was a relief, right? But that your model's not curing people when you're giving insults. But in these cases, right, there was some change of states. And then also you're seeing here where they ended up with and how many of them transitioned into that versus were there to begin with. So some are moving away, some are coming from different groups, and we're just kind of trying to represent that here. And then what we did was we went and tried to use different ways of predicting this state, whether it was at the 2 hour or the 20 hour, and also looking at sensitivity. So we used some random forest techniques, hypothesis testing. So we coupled the parameters, and in this also for the random forest with some of the readouts from the model, whether it was, the peak of the macrophages or the ratio of the amount of healthy tissue after 2 hours, just some metrics that you could pull from the transients combined with underlying parameters, and then did some just standard statistical things to see if they were significantly different across the different groups. I'm seeing some chats. Is that for me? Sorry. This is the main result from the random farthest, and you see that one of the important factors. So this is right, making a bunch of decision trees and determining which parameter is best at when you're forming these trees of correctly classifying things as whether it's moderate or severely damaged or healthy at the end of recovery. And I, again, cut off a table because it's just a nightmare, but these are not necessarily ordered for significance testing, but for random forest, they are ordered for importance. And you can see that different things came out as topper, on the list, but there were some parameters, I think you can see what I'm pointing to, that came out for all. And then EFAST is a global sensitivity method from Denise Krishner's lab, where it's not the classification, but it's seeing which parameters the model is most sensitive to at that time. And these were also similar to what was predictive of outcome. And so then this work is in a paper, and in that paper we spent time looking at using these as our target interventions. And so this KMNE here is collateral damage by the immune system. So whether that feedback loop of when you, not only does the ventilator cause damage, but then the influx of the immune cells causes damage. If that is too strong and creating a strong feedback loop, we think that that's kind of tied into getting you into that severe case based on some of the results we have here. So we were looking at that, and also baseline repair was a big one. If the underlying ability for those cells to repair, which may be something that differs with age or something to look at. So I may have talked incredibly fast because I can't remember the last time I gave a 10-minute talk, but I hope I have time to thank my collaborators and that I didn't gloss over everything and leave us with more questions than answers. And also shout out to my graduate student, Sarah Minucci, who's now at Applied Biomath.
Lovely. Reinhard, do you want to go next, or do you want to have one or two questions before you start?
We can do some questions, that's fine.
So there were a couple of questions in the chat. Ruchira and Gary, I think you had some questions.
So I think John's, I would need a little bit of time. Might you pick what you think are three of the best examples? I think I would probably have to take a few minutes.
That was a back channel to you because looking for content for this meeting in the fall.
Oh, okay. Got it. Yeah,
You
yeah.
have so thoroughly reviewed a large number of models.
Yeah, no, I mean, I apologize that I didn't realize it was a direct.
No, that's all right.
But
And
I just can't answer it off the top of my, it's a great question,
That's all
but
right.
I will do some homework on that one.
Yeah, I figure you're the expert on what would be the three best examples of using multi-scale modeling in the virus pandemic world. And let's put together a panel of those three papers and present some evidence. Thank you.
Nice.
Talk to you later.
Yeah. Gary, there's a current, we're moving to a new experimental model, and I had to, the initial stab at things, we had to add terms that were more associated with the volutrauma and the actual breakdown in the log, because we weren't getting, we think that some of the challenges in this is we're not really modeling the breakdown of the barrier enough to get that, it's still very diffusion based in this model. And so the next what we were, immediately we came up and we switched our context, and we're getting a lot more damage here where, it wasn't translating as well because we don't have that volutrauma. It's more of the, I would say the pro-inflammatory mediators produced by the barrier released versus the breakdown of the barrier letting influx in from the blood and that kind of stuff, and the resulting shunting.
Thanks. Yeah, I mean, this is, we've done a little bit of this type of stuff, focusing more on what some of the presumptive ventilator strategies might be, like APRV and that type of stuff.
Mm-hmm.
So, this etiology of whether it's pressure, stretch, or the opening, closing, forcing of it is actually the initiator of the injury. If you just start with the presumption of injury, then yes, that all works. But linking it to the biomechanical component is tricky.
And we with these older mice, we've had a lot of issues with them being able to handle the amount of fluid associated with the anesthetic. Because of their weight being so much more, these older mice were getting so much more anesthesia that it was causing volume trauma. I guess that's not the right word I want to use.
Pulmonary edema.
Thank you. So the grad student in biomedical, he actually switched gears a bit, and he has a paper related to, giving these old mice a different type of, anesthesia a different way, not to cause them so much unnecessary lung stress.
Interesting. I'm just going to post a link to a paper in case you're interested.
Yes, thank you. Of course.
I guess that actually.
So maybe. Oh, sorry.
I was going to say, Ruchira also had a question that was rather parallel, I thought, to the one that was Gary asked.
It's probably the same question if I understood Gary's question, but I don't know those words. But yeah, it was that is there some parameters of the ventilation, like pressure or volume or something that would be parameterized in the model?
Right now, that is no. But right now it is an epithelial subpopulation where you're transitioning from a healthy to damaged state proportional to, right now it's just everyone's getting the same amount of, source of damage. But you would expect that if it was a different type of ventilation and or different settings, that would give rise to different types of, even potentially different places that would feed into the model. Right now, it's just these cells become damaged, so they transition from a healthy to a damaged state.
Right.
Was
So
it- was
presumably
it-
if it's age dependent, then there would be different settings for different ages that might help.
Yes.
Maybe
Was
they already
it,
figured that out.
was it Ahmed Al-Jumaili in New Zealand who did quite a while ago, like 20 years ago, he was doing rather extensive models of ventilators to try to improve ventilator design to reduce injury.
Yeah, so, I mean, without going into Reinhard's time too much, I think a lot of the challenge during this entire period of time is identifying what the actual clinical etiology of this disease process is.
Yeah.
Right? So for a long time, the supposition was that if there was a high pressure that the alveolar sac saw, that was the thing that triggered the the damage. That turns out probably not to be the case. The next thing was whether it had to be a high volume with respect to stretch. That turns out that it probably is not the case. So now it is this opening or closing or the snapping open of the alveolar sacs, which is a more complicated architectural problem. So this is, so there's a biological component that Dr. Reynolds has been working on with respect to what happens once you get that particular thing. And then there's this whole biomechanical issue, which is actually an anatomic issue, related to this, because one of the great unknown controversies in pulmonary physiology is what is the thing that actually gets bigger when you take a breath? Because the very common cartoon that we have is this balloon that gets bigger and it opens and that kind of stuff, and that is not what happens. So it's a much more complicated spatial architecture than that. And it has relevance when you're dealing with different types of mechanical ventilation.
Great. So that's something we have. Let's dig into that in our discussion. That's great. Okay, let me move on to Reinhard's talk now. It's great to be able to introduce Reinhard. So I'll turn it over to you then without any more time. Let's see. Can you?
Thank you, James.
I think I have to actually do it this way. There we go. Okay, so Reinhard, you've got your splash slide, and now I will stop the share and turn it to you.
Oh, okay. All right. Thank you, James. What I want to talk about is something decidedly less glamorous than what we've just heard. We'll switch from the science of multi-scale modeling to something else. So we've, for those of you who've been part of this working group or stuck around, you've heard a lot this discussion about digital twins and building digital twins of the immune system and other aspects of this topic. And so James and I and others have talked quite a bit about finding grant funding. How much money do you need to build such a thing? It should probably be in the billions. And I'm always frightened when I think that somebody might actually go and give us $1 billion to build a digital twin. Then what do we do? And so we've actually thought about this for some time as part of an IMAC project that was funded by Grace's program that also involved quite a bit of science, but it also involved some thinking and work around, what do you need to build a digital twin and how do you build some of the infrastructure that goes into it. And I want to talk today about part of the work that goes into it. Now, we've heard in this forum quite a bit about digital twins, and it's pretty clear that there is no clear consensus as to what one of those is, what it might do. And so what I'd like to do first is talk a little bit about what I think of when I think of a digital twin, and then we can move on to talk using that particular framing of the issue. So let me just explain this. This is a slide or an image from a perspective that James, Jim, and I did a little while ago that outlines this view. And the view that I have is consistent essentially with the origin of this concept in industry. Essentially, you build a software instantiation of a particular technical device. The favorite one, I think, that most people use is an airplane engine, and then you customize that instantiation to the particular device with operational data. So in this case, for a medical analog to that, you would, let's just stick with, say, the immune system. You would collect together the requisite biology that goes into modeling an immune system. So this would obviously be a multi-scale model that you would need, ranging from intracellular features all the way to whole organism features. You would build a model like this. You would have many component models, most likely. You would validate this model, and you would most likely validate this model using animal models, using in vitro models. And once you have a generic model like this, so then we're on the the third instantiation of this figure. That's when sort of the next step of this process starts, and that is to personalize this particular model by calibrating it to data from a particular patient, so that you could actually assign like a social security number to that particular model and call it the digital twin of this particular patient. Of course, in this generality, it doesn't make sense to even talk about it. Digital twins would have a purpose and would probably, in almost all cases, represent some tiny portion of even the entire immune system. But conceptually speaking, this is what it would be. And once you have this digital twin and you've calibrated it with data from this patient, so that could be anything from information from electronic health records to periodic immune cell counts, to cytokine panels, to vital signs as the patient progresses, a number of things. And then you can use this personalized model to do various things. You can predict the trajectory of a patient in a particular condition, maybe an infection. You can predict or forecast, if you like, how that patient would respond to a particular therapeutic intervention, things of that sort. So this is the concept that I have in my mind when I talk about the digital twin. So it's two parts. It's really a generic model, and of course, many of us in this community build such generic models for a living, and then take an integration of these various generic component models into an overall a model that is then calibrated with data from a particular patient. So that's what I mean by a digital twin. Now, what do we need to build such a thing? So first of all, you need an appropriate team. That team will consist of biomedical researchers, it will consist of modelers, it will consist of software engineers, clinicians, maybe other areas of expertise need to be in there. So one needs to assemble such a team, and it needs to probably be a large team. One can argue, is this something that one can realize within an academic setting, or is it something that would need to be done in a commercial or industrial setting? But in any case, the crowdsourcing of models subgroup has thought quite a bit about precisely what it takes to assemble such teams and get them to work together. We're working on a manuscript that outlines the requirements for this and has some case studies that one can learn from. So that's one. The second one is you need to have a conceptual map of this generic model and a plan on how to validate it. It's, as James always likes to emphasize, this is one of the most important parts of modeling, and I completely agree. So that will be a very important one. And you certainly need a plan for personalizing this generic model to a specific patient. How would that work? What kind of data do you need? How do you integrate data streams? How does this all work? You next need an environment within which you build this model. You simulate it, you validate it, you test it, and you have the intended end users interact with it and test it. And you need an extensive information management system that keeps track of all these things. That, in my mind, by the time you're done, what you have is a medical device. That's how I think of a digital twin. It's a medical device. And as such, it will need to be approved by the FDA. And you need to do everything along the way that will allow you to get approval, keeping track of how you put this model together, what data you used for which parts, and what your various validation experiments were, will be extraordinarily important in that context. So to me, these are some of the key ingredients that you will need to assemble in order to do this. Now, what I want to talk about here, the work I want to tell you about here, is essentially the focus on the environment within which you construct this model. That's my part. We've also done some work on the information management system, but I won't talk about that today. So what are some problems that occur when you want to build complex models? One most immediate and important one is that by the time you're done building a complex model, there is no more transparency how that was ultimately put together. You have intertwined component models that depend on each other, that their simulation depends on each other, which makes it quite complex to follow what's going on. You might use incompatible data structures, component models, and incompatible languages. You might have an architecture that ultimately makes it difficult to extend models. You take something out, you put another piece in, and you try to run this and it breaks. And of course, you need an environment that makes distributed collaboration easy, because that is, in whatever form you do this, that's how it's going to happen. And these are the problems that we try to address with our work. So this is the paper that we published recently that I want to just very briefly tell you about. A modular computational framework. Now, of course, the concept of a modular model design and modular software design is not a new one. It's been around for a long, long time. And in one form or another, I think certainly most more complex models are built in this way. So we were thinking of a somewhat different approach that would address these issues that crop up when you build models like this. And that's what's in this paper. The paper is in part quite technical, and what I will do is talk about it in quite simple ways. We have Adam Knapp here. He's the lead developer of this whole framework, and he's certainly able to answer any more technical questions. So what we did in a nutshell is we built a hub and spokes kind of architecture. The hub consists of this gray box, which represents the global model state. So it's essentially a collection of data that describes everything about the state of this model at a given time. It describes the model geometry. It has all the parameters in it. It has all the data in it. And it is this model state that's the central object. The spokes are represented by the different modules. Now, in this case, a module is simply a computational algorithm together with some other information that is needed to run this algorithm. In particular, there are no data in any of these modules that is stored. The modules directly interact only with the global model state in the sense that they obtain data from it, perform computational operations, and then deposit data back into it. And that's it. There is no direct interaction between any of the modules. You see here, module 1 is blown up to show schematically what else is in there, but it's not really important for the purpose of the point I want to get across, is that this is the structure. So the modules have no data in them and do not directly interact with each other. To illustrate again the difference between the way things typically are done is in the way these modular frameworks are typically implemented is you have modules that contain both models and data, and that pass data and output from models to other models in other modules. So you have in the end, what comes out to be a substantially more complicated dependency structure. Whereas if you look at the bottom box in the design framework that we've developed is you have a very different structure, and that modules read and write from data fields. They certainly modify their own data field in the sense of modifying the data fields that describe the state of the part that this module represents. So this might be a module that represents the internal state of macrophages. So data 1 will contain the internal states of all the macrophages that are in this particular simulation, and it will modify those, but it could also modify the data fields that represent other modules and other biological entities, and that is how different biological entities in this framework actually interact, is by changing each other's data fields. Now, there's one other feature, and I don't want to go much into it because I don't have much time left. What this illustrates here is in the context of a use case of actually the immune response to a fungal infection in the lung, is how you might now add a new component to this. In this case, we're adding a module 8, which is hepcidin that is produced and secreted by the liver, and has various functions inside of the lung related to the immune response. It is exceedingly simple to add such modules, precisely because you don't need to modify the interface of this module with any of the other modules. You simply need to modify the data fields that that you're looking at. So that's at a very high level, in a nutshell, that is the structure. And I should emphasize that what this is not it's not a simulation platform in the sense of CompuCell 3D or PhysiCell or Copasi or anything like this. This is simply an empty software shell. It's written in Python, and it can be filled with whatever models you want to fill it with, and it does nothing further other than allow you to structure models that you integrate. And if you now look back at the problems that I identified earlier that can happen when you look at when you build complex models, they to a larger or lesser degree are addressed by this particular software architecture. In particular, this lack of transparency of model implementation is alleviated considerably. And I think from the point of view of building a really complex model, validating it, calibrating it with patient data, getting it approved by some regulatory agency, these sorts of things are absolutely crucial to get done. So what other things need to be done? One of the things we're working on, and that's important, is to essentially accommodate models written in any language. At the moment, the way it is right now is it has to be written in Python, otherwise there's a problem. But there are a couple of ways that we've been working on in which this can be done, both within the Python environment, and also we have an implementation of this architecture based on Docker containers, which allow you to run any model in any language, in any framework, and have these work together really well. This is, I think, from the point of view of getting a bunch of labs to help you integrate models, it's an absolutely crucial feature, because if you want to get somebody to provide a model for you and you say, well, you've written this model in C++, but actually I really need you to write it in Python. Yes, I'm almost done, James. Anyway, so my last slide gives the acknowledgements for the grants, and it also lists the team. And I mentioned at the very beginning that one of the challenges is to put together a team with the appropriate expertise. So what we've done here is we have Borna Merad in his lab. Borna is an immunologist who is also the division chief of pulmonary medicine here at the University of Florida. So this is the clinician slash biomedical scientist component of it. Then there is our lab here. We're the modelers. There are 2 graduate students at UConn that worked with me while we were there on this, and there's a bunch of software engineers at Kitware who have done tremendous work. In particular, helped us build a graphical interface and a lot of the software architecture here. So, but with that, I want to end. As I said, this is decidedly non-glamorous in comparison to the science, but I think it is absolutely crucial if you actually want to realize one of those projects.
Great. Thank you, Reinhard. So now we have time first for a few questions directed to Reinhard specifically, and then for a more general conversation.
Ruchira has her hand up.
Go ahead, Richard.
Thanks, Reinhard. So yes, that at least what you described sounds elegant. It reminds me of Gillespie's algorithm for stochastic simulations as far as the architecture. So as far as the digital twins, I feel that it sounds similar to something that at least several years ago, when I was familiar with them, this group that's a couple hours from you at University of, or actually Moffitt Cancer Center, University of South Florida, the Integrated Mathematical Oncology group, they seem to do. So that's on the digital twin side. And then as far as modular architecture, it sounded from Denise Kirshner's talk like they might have some kind of modular architecture, but they weren't necessarily doing digital twins. So so those are just some examples, and I'm sure there's many more examples of people in this group. So people who already have existing kind of frameworks that they've been using in their lab that aren't necessarily modular or don't quite fit the digital twin concept, or even, you know, these labs that I mentioned that are maybe partway there, how would they go about like reframing their existing work to fit, like how much work would it be to reframe such that it would fit into your architecture? Or would, I mean, it certainly looks elegant to, if one was starting de novo.
Yeah, it's an excellent question, Ruchira. So in a couple of weeks, we'll have Enrique de Assis present a model, a fairly complex model of the innate immune response in the lung to an Aspergillus infection. And Enrique has coded this in not in a modular way at all. And Adam Knapp is just about at the tail end of taking Enrique's model and coding it in this modular architecture. And if there's time in the discussion later on, Adam can tell us a little bit how much effort that is. But it's an excellent point. But it's certainly possible without hurting yourself.
Thank you.
Other questions for either of our speakers? I guess I can ask then one, since I've got Adam and Reinhard there. The idea of separating process from state is classically a very desirable thing to do. And so your approach is very interesting. One of the questions then is, where does the specification of whether a module is able to act on the data structure live?
Mm-hmm.
So you have to have some way of specifying what the minimum data set, what minimum data components are in the data set, so that a module is capable of acting on it.
Mm-hmm.
And that's typically, I'm breaking my rule of not asking a technical question during our canonical hour, but since no one else asked a question, I wanted to raise that one.
Yeah, since you expect the technical answer, I suspect I'll maybe let Adam answer that question.
Yeah, so we keep track of the model state. Each module lists a set of fields that it's going to add to the global data structure.
So it's sort of a frames-based approach to that.
Yeah.
So there was not a long answer. That's why I'm okay. Other questions, either for Angela or for Reinhard?
So let me just add one comment. In the paper, we have a detailed example of actually we take an agent-based model coded in NetLogo, and we rewrite it in this modular architecture, and indicate, for one, how you lose lots and lots of dependencies in the process. And by the time you're done, actually, you don't really know that this is an agent-based model. It's difficult to tell, but you see exactly how this process works.
And is there a specific simulation engine attached to the architecture itself?
Yeah, there is. So maybe again, Adam can probably say, give you the technical details of that.
So attached to the architecture, no. We have a few sort of generic things like a rectangular style grid that you can, attach, the fields according to fields of molecules on. And we have, a list of cells kind of class that you can use as a template, but it isn't necessarily tied to a specific implementation. We do have the original one that was in the paper and the model from Enrique de Assis that Reinhard mentioned that I'm coding up. And both of these have some similarities. They also have some significant differences.
Yeah, so I think it's fascinating work. I've been followed it for a while. I think the concept is fantastic. And I think when you run into ABMs is when these things start having trouble, because there's so many, as were alluded to, these software dependencies associated with the particular instantiation or the execution of these models that is dependent upon, how the thing handles schedulers, how the thing handles order, how it handles, sequence and these types of things, which are sometimes, specific to a particular software platform, even if you run your own, de novo model without using a toolkit. So, I think it's a fascinating sort of thing, seeing, and the hope is that it can, that multiple people can do multiple things with them. I think one of the other challenges associated with these types of unified frameworks or software toolkits is legacy models, right? And what is the activation energy required to port to a new system, maintaining fidelity to the original reference implementation when you do that particular port because of some of these software implementation dependencies. And then the lift in learning the new framework for people who are used to writing models in their own languages. Now, all these sorts of things are inevitable when any sort of unified framework is necessary, which I think for this particular scale of project, some type of integration is absolutely necessary. So there has to be a very large carrot at the end of this particular stick in order to get people to be willing to cast off their particular existing implementations and do it in this particular form. So when the argument with respect to doing this comes about, that is going to have to be part of the stick. That there should be a huge pile of money over here if you're part of it, and if you don't do it, then you don't get part of that money, and that provides you the incentive to take the extra effort that's required to port your models. And I thought Filippo has a question as well.
Yeah, I wrote it in the chat, but I might read it as well. My question is technical, unfortunately, and it is related to whether you have kept in mind this architecture for multi-core, multi-CPUs machines, because if this is, as I think, if this is the case, how you manage the problem of accessing memory at the same time when you have many modules running in parallel at the same time? Is this an issue, or you have a solution?
Yeah, so let me, Adam, you can answer that question as well, but let me just say two things. First of all, Gary, to your comments, they're absolutely well taken. But I think actually the biggest carrot we hope that's at the end of this is not so much a pot of money, but it is something that helps you do things that you might otherwise not be able to do as well. That's how I would see it. And Phil, to your question, so this framework is really independent of how you simulate. That's what Adam was saying. You can simulate this in many different ways, in multi-core, single core, and the complexity issues, computational complexity issues are, they are there whichever way you do it. What the advantage this framework has is that it has a very low overhead in the way it operates. One of the things that we had concerns about with a Docker-based framework is that it is computationally, that is just from the point of view of overhead, it's much more expensive than this is. But in terms of implementing it as a multi-core computation, it does not help you. It does not hurt you either. I don't know, Adam, if you have any other comments on that.
I could give a technical response. So the Python portion of this is single-threaded. Of course, we are making extensive use of NumPy, which contains some primitives which are, multiprocessor aware. The kind of simulations that we've been doing are typically stochastic simulations, and also we're running them mostly on Amazon Web Services nodes. And because they're stochastic, we are typically running rather large numbers of them so that we get a good sense of the distribution. And so we sort of are taking the view right now that, the sort of the memory bandwidth issue that you alluded to, they'll be running on actual physically different machines, some server rack.
So let me just break in for a second. We have the option now of splitting into groups or staying together. The topics were somewhat different. So why don't I do a quick poll, and if people feel that they would like to have time to talk independently with our speakers, probably the poll is basically I want to try to get a sense of, I don't want, if people have a lot of questions for Reinhard, and I want to make sure that those get answered, but I don't want people to not be able to talk to Angela about that wonderful work she was doing on ventilation injuries. So I created a poll where you could quickly tell me who you would like, whether you'd like to be broken out or like to stay in the room. Maybe I should just ask for a show of hands. Is there anybody who would like to be in a breakout room to be able to talk specifically with Angela and or Reinhard?
After the meeting.
Okay. No. Okay. Well, if people are happy talking as a group, that's fine. I just I wanted to give people the option. I don't want people to feel that they're excluded from asking questions. Okay, so I'll return the conversation to its interrupted flow.
So I do have a question. This is kind of big future. How do we envision this digital twin thing being used? Is it real time or is it offline? And the difference being, I gather up a bunch of data about a person and they currently have some medical issue, and I send data about measures of their medical issue off to the computer. And tomorrow I get a report that suggests where they might be headed. Or am I connected to the digital twin when I'm lying in the hospital bed? And when all of a sudden my blood pressure does something weird, the digital twin sends a message to the physician and the nurses that says, Mr. Rice's projection has just changed to he's going to die in an hour.
Why is there an or?
Right. How do we envision it
or?
being used? I'm sorry?
But John, why is there an or?
Oh, I don't know.
We've heard both of these presented already. The things that Angela was talking about could be used to optimize and understand better ventilator treatments. The heart flow people who raised $400 million last week are doing offline optimization and diagnosis with detailed digital twins. But as we know, things like artificial pancreas or defibrillators are using simple digital twins in real time to make this human out of the loop decisions. And so I don't think there's an or here. I'm interrupting because Reinhard should answer the question, but I'm confident he will say the same thing. There isn't an or here. It's an and.
I hadn't really thought about it until a couple of people were talking about computational speed and power and that's where it caused me to ask the question. Because I agree, using it for either one should be, but it's a big difference, I think, in demand on systems.
But let's maybe give Angela a chance to talk a little bit. If you were going to try to take these wonderful ideas and turn them into something which was going to be directly clinically applied, how would you start, and what would the lowest hanging fruit be?
So what we've done with the current like cohort, or I shouldn't use the word cohort with anyone clinical, sorry. The set of parameters, I got dinged on, was trying to predict where you would go. And if you didn't have confidence in that prediction, at least identifying within your parameter sets, when does that group dynamics clearly, right? Like if, for example, we took 20 of the parameter sets that had the closest similar outputs, and a lot of hand waving there, but we use some least squares type metric on just different types of variable combinations. If you withheld information from the local site versus if you had blood and local, would you pick out different closest type behavior? And if everybody, right, all 20 of them are going to be healthy, then don't intervene, you're going to be all right. If there's ambiguity there and in that set, right, 50-50, well, I don't want to make a decision based on that. But at a later time point, I see that I got a 80% confidence in a particular outcome, or that this is going to go to healthy or whatnot. So we were looking at if within the simulated population, is there a divergence in behavior at a time where then you could measure that individual again and see which trajectory you were on. Or you need to, and we haven't done this yet, that's why I was saying we had more questions and answers at some point, you need to penalize with if that's too late of a point to actually intervene, right? Like if you got to wait 10 days on the ventilator to see if the ventilator is going to do enough damage, not good, right? Like you need something where, it's still in enough time that you could actually intervene. So for us, it was, for a lot of this, being able to kind of, quantify the uncertainty into your prediction, and then choose a later time point to gather real patient data to supplement your simulated data.
Can I make a comment on that answer, because it made me think of something which I think would be interesting if you were to look at this, because there is no treatment for this process. The ventilator is mandatory in most circumstances, and therefore, and it's controversial with respect to the specific mode that might reduce the injury.
I think this would be, maybe not, this whole concept is more, maybe not just ventilator, but
yeah. So I think the thing though that would be very cool is if there could somehow be tied to that high risk group of, like, let's just take COVID patients who are getting non-invasive ventilation prior to intubation, and you're in this kind of cusp of deciding whether to intubate or not. There might be a certain subgroup of patients that have higher susceptibility to the mechanical ventilation as opposed to non-invasive mechanical ventilation type of thing, just as a inflection point to potentially.
I mean, I always like, if we could find something with the initial condition, right, and not have to, if those were indicative of end state.
Also, I think,
Well,
I think one of the things that is cool about this, as opposed to many, more non-mechanism type of models, is you aren't completely dependent upon T0, right? Because you have a trajectory, you have a dynamic process, and as long as the model itself doesn't presume that there's no perturbation pre-existing at T0 with the simulation, you're just trying to find some period in the dynamics of that particular model in which you can, and let's assume for a second that it's, that the hysteresis is not super important at that point in time, that you can propagate forward from whatever time you happen to select, then you just go from that particular point. This is a huge issue with respect to all simulations of sepsis, which, if you try and tie to clinical data, you have no idea what T0 is, and therefore it's irrelevant at some level what T0 is. You just need to know that from this point on, some damage exists, and therefore it propagates in this particular fashion and then fit to your model within that particular forecasting potential horizon. So,
Yeah, so I did sepsis in my graduate studies, and when I came to VCU and they wanted to do wound healing, I was like, whoa, you know T0. Well, I'm on board. Like,
yeah.
you know when you got hurt. Okay.
Angela, you mentioned you were working with a biomedical engineer. Can you just say a few sentences about your experimental system?
So she has two different ones we've used. So she has a lot of times she starts with a cell line for alveolar epithelial cells to do some, proof of concept cells, but then she tries to use cells that are from the primary source, from the mouse, and then they're plated, and she has a stretch wells that are stretched radially, and she stretches them at different levels to mimic normal breathing versus ventilated, there's I guess a whole can of worms there as to what setting is equivalent to normal breathing and what is equivalent to ventilation that I'm not getting into there. But so she looks at stretching them at a low and a high stretch, and then taking the media off and recording the pro-inflammatories and this type of stuff. And we've done some primary source cells from old mice, young mice, and then also done some, media from old mice given to young cells and these different kind of combinations. And then on the animal side, she has ventilation protocols where originally we did it was a set tidal volume, and now it's been switched to pressure because of how we had so many issues with the old mice. The old mice really do not do well in these scenarios. And right now those are mostly vented for either 2 or 4 hours, and the next round of experiments we're trying to get some recovery in there. Also some mice that are then post ventilation are not sacrificed, and there they do the BAL. I'm drawing Gary can probably correct me on what that actually stands for at this moment, for the mediators and
The alveolar lavage.
yeah, thank you. And some of the phenotyping of the immune cells and stuff. So a lot of cell-based stuff. When we, first came at some of this with AB, and then when we moved, we're trying to move more to having both blood measurements from these mice and the ventilator itself. The device can do these periodic testing to get some recordings on the lung mechanics. So we want to be able to incorporate changes in the lung mechanics as the ventilation is going on, right? It can stop the ventilated breath and do some controlled breathing type things and get some readouts. So we want to kind of combine that as a metric for lung health, along with these more end state things that you have to sacrifice the animal to record these cells and things. But so combining the blood to have some time series, lung mechanics time series, and then a lot of these end state. So that might have been way more complicated or not as experimental. More
That's
what I
actually-
get from them.
I mean, you answered a lot of what I was going to ask, which is, if we imagine a patient in the ICU on a ventilator, what information on patient state do you have? I mean, you have some blood chemistry from time to time. You have blood oxygen level. You have heart rate. You have tidal volume.
We also
what are the metrics that you use to characterize patient state that would be easily available, and what ones would you like to have that maybe are not so easily available?
Yeah, so we recently got funding through a Virginia, it's a Virginia-based grant. It's the Commonwealth Cyber Initiative for a lung sensor, and the idea we're working with engineers now in on the security side of it, so that they're designing a sensor that can send data and not be hacked and stolen. Data can't be stolen from it, but the original goal of that was to more make it work with something that can be read out from an ICU patient, whether it's from from their blood or from their what do I want to say? I can't think of the word right now, but sorry.
Reminder.
So
Your monitors, the mechanical ventilator has a great deal
or the stuff that comes out of the ventilator when they're breathing out.
The expired gas secretions. All that.
Yeah. And so this is kind of like a more of our we you know, a little bit of a sci-fi kind of moment in our original, like sitting around the table and stuff, was if you had this sensor that could at least pick up on a few of your mediators and just give an alert to, you know, kind of go do a better test or go do something else. And so we're kind of diving into that and thinking, you know, that in terms of the model, like you're saying, what would you need to know? And right now, a lot of the times is, you know, we're going to be confined to particular things. How often do you need to know them? You know, is I think our first question we're going to tackle. But
Hey,
I mean,
Angela, kind of a naive, like non-doctor question. Would
I
you,
have those all the
what,
time.
yeah, what difference would you expect to see if the patient had mild COPD versus a patient that came from the Andes Mountains? What would you, I mean, would you start out assuming that those two patients are going to react the same to any given combination of whatever you do with the ventilator?
No. I mean, I think that the COPD itself, right, is right. Your underlying lung tissue is damaged. There's probably already an inflammatory response going on there, you know, so how that underlying inflammatory state makes you already have damage, and then you're adding another damage mechanism to that.
But presumably the digital twin would know all that, because if it didn't know that, then it couldn't figure out what was going to happen.
Yes. So
it would also know that I came from the Andes Mountains and arrived here like last month.
Yeah,
the twin would have to know that.
Well, the twin, well, all right. So I think one of the challenges of the twin analogy is it presupposes that you have what an equivalent of genetic identity and knowledge about how that translates into change in the function. I think in terms of how these things are probably going to work is, as Reinhard discussed, there's probably some underlying specification that we assume is consistent across all human beings. And therefore, there are different parameterizations associated with that specification that determine the responsiveness or dynamics associated with a particular instantiation of that specification. And we don't know necessarily how an individual fits on that particular thing, and it's probably not going to be consistent over the course of their disease because people change, right? So then this is probably more nuts and bolts, but the calibration of a particular digital twin to a particular individual is almost going to necessarily have some sort of burn-in time, right, to measure some time series and hopefully some sort of non-dangerous perturbation that can help delineate what the responsiveness of that particular individual is of that particular system to a particular perturbation. So you'll essentially have this model that, you know, theoretically can represent those particular dynamics, you know, start measuring at a particular point when people start getting sick, right? And you say, okay, you're going to measure again in 30 minutes. All right, and if the change is this way, then it constrains parameter space in this particular fashion. You have a intervention, some sort of non-lethal, non-pathogenic sort of perturbation that says if this thing happens, then this should respond in this way. If it doesn't, then it's this particular trajectory. I mean, there's this sort of burn-in time that's going to be inevitable for the initial implementations of these types of systems, because you know the artificial pancreas isn't necessarily tuned to an individual right away. It has a calibration period to determine what your particular response to insulin happens to be, right? And it continually updates what that particular relationship is over time because it has the input and the output. So that some sort of variant of that particular process is going to inevitably happen. So the hopeful idea of this sort of thing is it could be done and we're talking now almost exclusively about some sort of acute illness type of manifestation of digital twin, because that's the motivation of this particular working group, is that there is some time sensitivity with respect to that, but there's also a rapidity of the expected process that's happening that says that this process happens quickly enough, and hopefully the simulations will tell you what that sampling interval is going to need to be able to help start locking down what the potential parameter space that you have to be operating in, and that's going to be refined as you propagate forward in time. But you aren't going to have, in most cases, pre-morbid, pre-disease values of anything, because we don't measure cytokines in people walking around. And even things like heart rate variability and these types of physiologic signs, they are, global aggregate measures of health. They do not necessarily specify what happens to be wrong with an individual person. So conceptually, if we're talking about acute illness, it is going to require some sort of burn-in period from the initial application of whatever sensor array you're going to use to calibrate your digital twin to that particular individual during that particular time. So this falls in the category of your real-time implementation component, right, which is a sense, prediction, forecasting, actuate, potential control type of thing, which is way down the road, right? But conceptually, that's what it probably look like. The more tractable sort of things are the sorts of things that Angela has been talking about and a lot of us are talking about, is building virtual, and Filippo's talked about this too, where you build virtual populations that represent some degree of population heterogeneity, and you identify what their particular characteristics are, and then you can potentially use that to identify various control policies that might be tied to identifiable components that are potentially related back to individuals, right? I mean, so that's like the in silico trial type of thing. But, I think the digital twin at the moment is a very useful nomenclature, because people listen to you if you say that particular word. I think we have enough flexibility with respect to what actually that means, given the very broad translation from the industrial application is, and some of the issues that Reinhard talks about with respect to integration of knowledge towards these types of things is part of that particular process. On the other hand, there shouldn't necessarily be a supposition that all the digital twins that operate within an ensemble of a digital twin in this array of things that are attached to a particular individual or in that discovery process necessarily have to be implemented in the same particular platform, just as the various weather prediction models that are used are not necessarily all the same instantiations or implementations or even same platforms. I don't know the details, but I don't think there's a great deal of interoperability between the American weather system and the European consensus weather system. They are just equally valid models that run continuously in parallel, and I would expect that there would be an ensemble of models that would serve the same sort of purpose should this thing ever get to clinical relevance, that there's going to be, multiple simulations or whatever of these people that are all taking information, forecasting, and they may have different capabilities. And then there's a separate integrative level that's going to need to establish what constitutes consensus, consensus about this. And I didn't read this thing. Yeah, that's right. So this thing, we've talked about this before, and this is the actual power with respect to the global idea of moving towards this concept, is that NOAA, right, is able to mandate, if you want us to use your weather systems, your weather prediction models, these are the things that you have to have. Conversely, I think we have to almost flip that particular switch and say to people, if you want X capabilities in the future, right, being able to prognose acute illness, if you want to be able to provide multimodal adaptive control, right, in an acutely ill patient, to potentially titrate ventilator concurrent inhaled anti-mediator therapy for ventilator-induced lung injury, right? You are going to need to generate the data in XYZ form, because the only way you're going to be able to answer those particular questions is with simulations that have these capabilities. I think that is the hard push that we have to have, and the argument that we need to make with respect to this. Right? We've talked about this before, but I think this is model-driven data collection, right? And I think the challenge is to make the case strong enough that the necessary requirement for these particular capabilities are the types of models that we're talking about, right? And we've talked in circles around that. That's an open question. We seem to believe that that is the case that we need to make.
Wonderful comment, Gary. Other questions, comments?
I have a
We have
question.
about 2 minutes left.
One more question.
Go ahead, Gary.
Are you working with anyone who's using smart dust or micro sensors or stuff like that?
No, I'm going to write that down though.
Yeah, right. Because that's something, when you were talking about, sensors with respect to what the composition is of the secretions in the lung, measuring at the level of the endotracheal tube is difficult because it's an aggregate of a whole bunch of stuff, and that's actually not where a lot of the biology is happening.
Yeah.
So if you were able to give inhaled particles that had sensor capability, then you actually get down into the alveoli to see whether or not you can, sense things down there. So
no, thanks for
you might
that.
want to ask your biomedical engineer about.
Yeah. Thank you very much, Gary. Thank you all, actually. It was a great discussion.
Okay, well, thank you everyone for staying on. It's great to have had such a stimulating pair of talks. Please help us out filling the schedule. Angela, if you have other people in your group or your extended group that you'd like to think we should hear from, lung injury and recovery, fibrosis are very interesting to this group. So anything on fibrosis and on the experimental methods used to get the data set.
I'll send you some information on my collaborator on her side of stuff. She does a lot of different. She does a lot of different lung what do you call it? Lung diseases and stuff.
I mean, sensor technology is also potentially very important. The fact that you could get information about the composition of the exhalate when you're attached to ventilators gives you access to things that are important. So
yeah, I mean that's part of the sci-fi part, but right now. But yeah, that's what as Gary had mentioned how hard it is. But
okay, well, thank you everybody. Thank you, Reinhard. Thank you, Angela. We'll see many of you tomorrow at the steer group, and look forward to talking with everybody in the next few days.